Okay guys, my name is Mr. Mpepu and today's date 20 September 2024 and the time there 51 minutes past 11. With that being said guys, Anbonani, Dumelani, Afsheni, hello. Guys, I personally welcome you to our Life Sciences Grade 12 revision classes, Term 1, Week 4 to Week 5 today. So thank you guys for joining in. Thank you for tuning in. If you are new here, don't forget to subscribe, don't forget to like, don't forget to comment, and don't forget to share, guys. If you're a returning student, thank you for pushing this movement together. So thank you. Don't forget to like also, guys. And then today we'll be done with meiosis, hopefully in less than 10 minutes or 10 minutes. So then we'll done with our abnormal meiosis and consequences, and then the similarities and differences between meiosis and mitosis in less than 10 minutes, guys. Hopefully we'll cover this. So thank you for being here. Thank you for choosing me, guys. Thank you for tuning in. Yesterday we managed to reach this slide on our classes. So if you are missing them, guys, don't forget to go back and rewatch, and then if you haven't subscribe so that you don't miss other videos. And then we start with the differences between meiosis 1 and meiosis 2. We did this yesterday in fully, so today we are only just going to do a revision about this. Meiosis 1 and meiosis 2, what is happening? The difference is that here we see that they are the chromosomes, they arrange at the equator in homologous pair, whereas in meiosis 2 they arrange individually. The chromosomes, they arrange individually, and then from there the whole chromosomes will move to the opposite pole. But this side we see that the chromatids, because one chromosome was like this and then it was divided in two, we see that now the chromatids are the one that are moving to the opposite poles of the cells. From there, but two cells are formed at the end of this division. We start with one cell and then result with two cells, and then this side four cells are formed at the end of this division. We start with two cells and then we end up with four daughter cells. The chromosomes number is halved during meiosis from diploid to haploid. Here they start with haploid and then they end up in haploid. And then this side we see that the crossing over takes place, and then in meiosis 1 there is no crossing over taking place, same as mitosis this side, right? The differences there, they are also same as mitosis. And then from there we can compare between mitosis and meiosis, Mr. Pep. Mitosis only occurs on our body cells, but meiosis occurs in our sex organs, our testes and ovaries. And then both chiokinesis and cytokinesis occurs once in mitosis, but in our meiosis, because it's two-stage division, we see that two or both of these chiokinesis and cytokinesis, they are occurring twice. And then this side where the two daughter cells are formed, and then this side the four daughter cells are formed at the end of this cell division. The daughter cells are genetically identical, but in this side they are genetically different. All these four of them, they carry different genetic material. And then this side the chromosomes number remain constant. If we start with 46 chromosomes, we are also going to end with 46 chromosomes. But in meiosis, that is not the case, Baba. Chromosomes number is halved. We start with 46 chromosomes and then we end up with 23 number of chromosomes. But crossing over does not occur in mitosis, yes, but in meiosis crossing over occurs during prophase 1, to be precise. But the only similarities between mitosis and but the only similarities are between mitosis and meiosis 2, right? That what I was talking about. In mitosis we have prophase, metaphase, anaphase of only chromosomes separating in our centromere. Also in our meiosis 2 we see that what is separating there is our centromere being pulled by those spindle fibers. But the difference between mitosis and meiosis are illustrated in the diagram below. As you already know or you understand what is happening with our mitosis. In our mitosis, as we can see on our left hand side, we have prophase there, and then the chromosomes, they are not paired on the homologous pairs. You can see they are single, and then from there we move to the metaphase, where we see that precisely those chromosomes, they are single arranged at the equator. From there the anaphase is going to pull those asks those chromatids to the opposite poles, and then the telophase there, the cytokinesis, is going to split that cell into two identical daughter cells. And then from there we move to our meiosis. In meiosis there's different, and then the two cycles of our cells that are being divided into two, meiosis 1 and meiosis 2. In prophase 1 we can see that the homologous chromosomes pair up. Then this one, they are preparing for bivalent, and then they're also preparing for crossing over, right? The process that happened in prophase 1. And then from there we move to our metaphase 1. In in metaphase 1, the chromosomes pairs line up in the equator. So now they are in pairs. So there will be 23 pairs, 23 pairs, right? And then from there we see that after here it's also after crossing over. Then from there we move to our anaphase, where a whole chromosome is going to pull to the poles. And then from this you can see from this illustration here, a whole chromosome with 4 chromatids to be precise is being pulled to the opposite direction. And then in telophase 1, don't forget that the cytokinesis is going to take place, and then 2 cells are going to be divided. Those 2 cells that are divided, they're also going to undergo a second meiosis in prophase 2, guys. As you can see that this phase here, it's not different with our phase here in mitosis, right? A single chromosomes, and then the difference between these two is that you can see that there was a crossing over that took place there, there's a crossing over that took place there, and then these 2 chromosomes now, and then these 2 chromosomes share the DNA from both parents, right? And then as you can see, the meiosis 2 take place, metaphase 2, a single chromosomes, they are arranged at the equator. And then from there, anaphase 1, the chromatids, they are being pulled same as this side. And then from there, the telophase, as cytokinesis, it's happening. It's just that in this side it's happening twice, or it's happening on 2 cells, and this side is happening only on 1 cell. And then at the end of the day will result with 4 haploid non-identical genetically cells. That was mitosis and meiosis in nutshell, guys. So we can move on to activity. My timer, I didn't do a timer, guys. Let's move on, guys. Okay, let's move on. But various options are provided and possible answers. Okay, which one of the following correctly describe the daughter cells produced by meiosis? We produce meiosis, we produce haploid, yes, and then we also produce them being different there. But if there are 28 chromosomes in the body of a donkey, how many of these chromosomes are autosomes? We're only going to minus 2 here, and then we're left with 36, because those 2 will be the sex chromosomes, right? The gonosomes. Use the scratch below to identify the process 1, 2, 3. What is happening? Process number 3 is producing zygote. Process number 1 is producing multiple cells. This one will be mitosis, this one, right? Yes, mitosis, meiosis here, and then fertilization here. And then cytokinesis is the term that describe what? The division of cytoplasm, where number B, there's the plasmic division, right? Is the term cytokinesis. And then the nucleus division, karyokinesis. The diagram below represents the 6 diagram phases of meiosis taking place in particulars in a particular cell. And then the diploid number of chromosomes in this cell is the diploid number of chromosomes. How many we can count here? Number 4, 1, 2, 3, 4. As we can see, we have 4 number of chromosomes, right? And then the correct sequence from the start of meiosis till the end is, we'll start with number 3 there. As we can see, they are arranging, or whether they are arranging at the equator or they are condensing. And then we can see number, from there they can be pulled on the opposite direction. Then will be number, let's see, it will be number mark. Hey, you'll do these activities, guys. Hey, I don't have time. You do the activity. Interface is the stage during which, comment there, guys. Interface is the stage during which phase. Interface is the, I'm disturbed now. Interface is the stage during which nothing happens in the cell. No interface, a divided cell forms, no cytokinesis, a cell growth, and the DNA replication, right? So let's move on, guys. You can do this activity, take place to form sex cells, meiosis there. You can choose, guys, what results of genetic variation, crossing over, and random arrangement, they result in genetic variation, both of them. Study the diagram below. You can see this one diagram. You can see, or we are pulling our chromosomes to the opposite direction. As you can see, the crossing over is already occurred. But what type of cell division is occurring? The cell division that is occurring, it's meiosis, guys. And then we move on. They didn't ask the phase, and then but what phase is depicted? It will be anaphase 1, if I'm not mistaken, right? So we do these activities. I didn't do my timer there. That's the problem. Refer to the diagram below, which shows the cell division of meiosis. There you can study the diagram. But abnormal meiosis, chromosomal mutation, we have gene mutation and we have chromosomal mutation. And then this mutation we see mostly in anaphase 1 and anaphase 2 due to non-disjunction, guys, of chromosomes, right? There's non-disjunction. And then we in our human what cells we see mostly suffered from a chromosome pair number 21 in humans being multiplied or not disjunct. And then we can see chromosome 21, the trisomy, and then Down syndrome. The abnormal is Down syndrome. They like to ask this question very much. And then you can see now the non-disjunction that is taking place, and then we have non-disjunction this side, and then we'll end up with these 2 cells having one extra chromosome. Same as this side can happen in anaphase 2, where that non-disjunction is going to happen, and then we see that only one cell it will have the double, and then only one will have nothing. And although this side if it happened in anaphase 1, both two cells will be having two short of one chromosome. But if it happened in anaphase 2, we see that only one cell there result in no chromosome. And then enrichment, this is the Down syndrome, as you can as laibona ngwana tlhapiwa Down syndrome. And then Down syndrome we see mostly in ovum, where one chromosome, as you can see, non-disjunction happened there, and then one chromosome was supposed to be this side, and then it end up in this side, and then also it's going to happen here, and then these cells there, that cell there, or that ovum there, is going to have an extra number of chromosomes, and then is going to fertilize with these 23 chromosomes, and then we end up with more chromosomes in pair number 21, right? And then there they can see they give us the this mutation leads to various malfunction in development of chest, upwardly slanted eyes, small nose, small mouth, and various degrees of mental retardedness, decreased muscle tone, hearing loss, and the heart defect, the change of non-disjunction. As you can see, guys, blah 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 blah. The parents can then decide whether or not to have a baby. Yeah, if we check you and see that you are baby, if you are pregnant and then we check and we find that you are baby with Down syndrome, you can decide to kill that baby or grow that baby and give birth to that Down syndrome baby. That's what they're saying up there. Then there's the activity, end of activity. Today's Friday. Thank you guys. I will meet you again next week. And then thank you guys, and then we can do this in if 1 minute, if also during which phase you can do this on your own, guys. This is an activity. Which one of the following combination results in genetic variation, meiosis, sexual and mutation there? And then you can do this on your own, guys. And then the biological term, but there's the biological term, a phase in a cell cycle that occurs before cell division, the phase in a cell cycle that occurs before cell division interphase, and then a source of genetic variation that arises during metaphase 1 crossing over. So you can do these activities, guys. Chromosomes align at the equator. Metaphase 1, metaphase 2 occurs during telophase of meiosis. Division of cytoplasm, no. Centromere move to the opposite poles. Occurs during the telophase, yes. Division of cytoplasm. And then we move on, right? Do that activity. As you can see, there's another activity there, and then with this activity you can see what is happening here. We have our crossing over that is taking place. This one will be prophase 1, and then we can see the chiasma there, and then we can see the centromere there. Mostly this question will be about that. And then this one we can see that we can arrange this from C to B up until to A, right? Those are the question arrange. There's the question, arrange letters A, B, C to show the correct sequence of the event, right? Then we move on 1.5, another activity. As you can see, we have all these cells taking these forms of cell. You can see here we have cytokinesis taking place, telophase 1, hopefully, yes. And then this one here, we have our telophase 1, anaphase, telophase 1, guys. Sure. Or are prof and these guys. So guys, do these activities, guys. Label structure W and X. Is just that I'm tired. Label structures W and X. Where are we? Cell membrane, and then homologous chromosomes, and then how many chromosomes are present in each cell. Phase A, where is phase A? 1 2 3 4 1 2 1 2 3 4, yes, 4. And then phase C 1 2. This one will be 4, this one will be 2. Give the letter of the diagram that represent anaphase 2. It will be anaphase 2, the number B here. Number D there, sorry. Number D there, as you can see that a single chromatid, they have been pulled to opposite directions. State the name and function of region Y and Z. Y there, centromere to join the chromatids or to join the spindle fiber. And then number Z there, spindle fiber to pull our centrosome or our chromosome to the opposite pole. And then in this specific there, to pull those chromatids to the opposite pole. Which phase precedes or occurs before phase A? Yes, precedes, yeah, before phase A there. It will be interphase, right? The one that we don't talk much about, it the preparation of a cell. And then that one will be prophase vele. And the diagram below represent the phase in meiosis. Cell Y undergoes division to give rise to cells Z and is X and Z. Some alleles are indicated by the letters. As you can see, there will be R multiplied by capital letter T, small letter t. You still remember this, right? And then but explain why cell wall does not belong to a human because of 4 chromosomes, right? Here we only have 1, 2, 3, 4 chromosomes, and then in a human being we have 46 chromosomes. Then how many guys, you'll do these activities, right? There's the activity 2.2, study the diagram below, represent 2 phases of meiosis and answer the question that follows. Identify, as you can see, let's identify diagram 1. You can see that one is metaphase 2, right? And then in diagram 2 you can see that one is will be anaphase 2, and then there will have non-disjunction, and then B will have our chromatid, right? And then we identify the phase represented by diagram 1 metaphase 2, diagram 2 anaphase 2, and name the part B. Where is the part B? Chromatid, to be precise. Chromosome, they might penalize you. And then describe, it's a chromatid, right? Describe what happens during the phase illustrated in diagram number 1. In diagram number 1, describe what happens during the phase illustrated in diagram number 1. What happened during metaphase, right? The chromosome, they are going to align here, facing the chromatids facing any other what we call, and then the central, the spindle fiber, they are going to attach to the centromere there, and then something like that in between. In diagram 2, the part cycle and labeled A is abnormal during the process of meiosis. Name the abnormality. Down syndrome, right? What genetic disorder could result in human if these abnormalities occurred in chromosome pair number 21? What genetic disorder? The genetic disorder, you will have you have pair number 21 chromosomes, right? And then from there, give 1 symptoms of the genetic abnormalities mentioned in question number 4. Memory loss, hearing loss, right? We did them up there. Hearing loss, small mouth, small nose, blah 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 blah. The activities, they're still there, guys. Oh, you'll do this. Describe the behavior of chromosomes during the process. For instance, one, prophase 1 metaphase chromosomes homologous, they're crossing over here, crossing over in pair, my equator, and then a chromosome, the whole chromosomes to different poles, and then cytokinesis. And then 3.2, as this diagram, X, Y, and Z crossing over is taking place, and then we have homologous, and then we've already done here, give the labels of A and B. Where is A? Homologous chromosome. Where is B? Central. So you can see that the question are repeating itself, guys, so that you'll have to understand to differentiate the two. You will be able to write them down. You will be able to understand, not to just memorize them, but if you are writing them down, if they're asking you a same question in different context, guys, it makes you to understand, guys. It makes you to understand. And then the last question, explain why nuclear O and P do not have chromosomes, because what we call non-disjunction happened, right? And then name and explain the disorder that will result in diagram M. Represents an egg cell that fused with an abnormal sperm cell. Guys, thank you for being here. Thank you for choosing me. Thank you for tuning in, guys. Until we meet again, we'll be starting with our new strand next week, guys. Thank you for being here, guys. Thank you very much. Until we meet again, guys. Today we said time. I didn't do my timer, but hopefully you'll agree with me, or you will you will accept my apologies, guys. Thank you. Until we meet again. Peace. Bakaina, those who like to uchela nenge, uchela nenge, guys. That's my number. That's my, I so bonely 10 rand, but it's fine, guys. It's fine. We are doing this to help our brothers and sisters out there. So thank you for being here. Thank you for choosing me. Thank you for learning. Thank you for loving life science. I love you too. Danko.